This video abstract is AI generated. How does an enzyme know which temperature it is supposed to work at? Homologous enzymes can share almost the same structure, yet work best at very different temperatures. In this work, we asked how sequence encodes this thermal tuning in the C-terminal domain of bacterial enzyme 1, or EIC. EIC contains disordered catalytic loops that must sample compact, active conformations for catalysis. Mesophilic and thermophilic EIC homologs have the same overall fold and conserved catalytic residues, but their loop sequences and temperature-dependent activities are different. To isolate the role of the loops, we used mesophilic EIC, thermophilic EIC, and hybrid enzymes in which the catalytic loops were swapped between scaffolds. This showed that loop sequence is a major determinant of catalytic dynamics and activity. We then used Hamiltonian replica exchange molecular dynamics to sample the thermophilic EIC loops across a wide range of effective temperatures. For each temperature, we calculated how often pairs of loop residues contact each other, generating a temperature-dependent contact frequency matrix. Principal component analysis reduced this large contact network to the contacts most sensitive to temperature. PC1 captured the dominant effect of heating. Many contacts break as temperature increases. Later PCs captured subtler behavior, including contacts that form or stabilize over specific temperature ranges. When these temperature-sensitive contacts were mapped onto the structure, they formed clustered dynamic modes within the alpha-2 beta-2, alpha-3 beta-3, and alpha-6 beta-6 catalytic loops. This means that temperature does not simply melt the loops randomly. Instead, it reshapes specific contact networks. The key test was mutagenesis. We introduced mesophilic substitutions into thermophilic EIC at selected positions and measured PEP hydrolysis activity. The strongest activity enhancements came from mutations at the most temperature-sensitive positions, including M351, Y301, F295, and L334. Together, these results show that disordered catalytic loops act as tunable dynamic elements. Temperature-sensitive contacts provide a molecular code that links sequence, loop dynamics, and enzyme activity. To know more, read our work online.